we go a long way with Philippe, and I'm very pleased that you have this opportunity to work, in two different countries so very closely, and thanks to David and Felipe and all the crew to organize this, and Carlos and the rest of the team. And so what I'm going to talk about, as Philippe mentioned, is in a way is a continuum of what other speakers talked about, and it's very interesting. I don't know if you guys thought about that logic, but we are kind of getting more and more broader in terms of biodiversity and organisms, and I think we get ultimately to where diversity has not been very well understood in the microbial one in the next one. So I'm not going to talk so much about microbial ones because you're going to hear, but like starting from genomes and assemblies, going towards non-models, and now we're going to talk about biomonitoring and biodiversity a little bit further. So I have, I don't know if you see this, but this is a map of Earth at night. And I'm interested in urbanization and how it changes biodiversity. So if you really want to have a map of where things are in terms of urbanized areas, this is a good one because you can see everything. But this is more of an inspiration for me to see something like this and put it in the context of biodiversity. And so, and arguably, I would like to argue that DNA information can provide a map of biodiversity for us. And this is, as you have seen and heard, it's possible to use DNA to understand biodiversity in different groups of organisms and different contexts. But the challenge, which I think, I've been thinking about a bit, is how to use a map like this to monitor changes in the environment and the ecosystem over time and space, specifically over time and space. It's in a way, like we started having maps long time ago, but then we put them on a system that we can have in our pockets, like a GPS, and map positions of ourselves in the globe. So that's the idea. So the data generation aspect and analysis, in my opinion, needs to change so that we can use this to monitor changes. And that's where I think that next-gen sequencing technologies have revolutionized this field, and I'm going to talk about that. So what is biodiversity? There are so many different definitions. This is the one I like, that from Ed Wilson, is, in a sense, is everything. Every living thing, every living organism and the molecules in them sort of represent biodiversity. But then a more pragmatic one is that I sent an email to my colleague, and I don't know if you see him. This is him, not the cow. But is Dan Jansen, the father of tropical ecology and conservation, and the one that actually kind of is coming from the same time of Ed Wilson, but he's been in Costa Rica and trying to conserve biodiversity. And I think his definition of biodiversity is a bit more complex and perhaps a bit more pragmatic. So the biodiversity of a place, country, county, habitat, ecosystem, a square kilometer, is the crude sum of all the species who live there and their genetic as well as morphology, physiological characteristics, and their interactions with each other and with their physical environment. However, of course, once you move away from the simple list of species, the task of measuring biodiversity as a whole becomes truly enormous. This means that when you move away from the species list, it's normal to think about like biodiversity of place or interactions, like plant defensive chemicals, trophic webs, or mutualism, and etc. So this has been a challenge, we really have to focus even within the context of biodiversity. It's very difficult to understand it as a whole. Now, if you look at a diagram like this, like a word cloud of the definitions of biodiversity, what comes out is things such as the species, varieties, in the context of ecosystems, and then communities and variability and those type of things. The species sits in the middle, kind of the largest sort of object for biodiversity, but we need to remember in the context of ecosystem, and also looking at, we saw in the last talk that how population variation can determine some of the conservation efforts and how genomics can help that. So unfortunately, we haven't done a good job so far of documenting biodiversity. This is kind of what we know about biodiversity, about 1.9 million species of plants and animals. I don't even have microbial ones and some more like protists and things here, because we really don't know much about them. And even these, they don't come like in a central registry, like, a library that you have all those. And in fact, I just before this, I was in EBI in Cambridge, and they were discussing, projects like Catalog of Life, Species 2000, Encyclopedia of Life. So these are attempts that just started to document biodiversity. Now, perhaps a big challenge is even the difficulty in understanding or estimating biodiversity, and these are the type of estimates we have that are really rough. And in fact, recently there was a paper, I don't know if you noted, in PLOS Biology, that was a bunch of colleagues a group of colleagues in Halifax. They came up with a number, 8.7 million species. But I don't have that number here because I don't think that's so much important. What is important is what I think they mentioned in this code, that unfortunately limited sampling of the world's biodiversity has prevented to date the direct quantification of the number of species on Earth, while indirect estimates remain uncertain due to the use of controversial approaches. So we don't have the tools to understand biodiversity. And here is, like when you want to view, maybe I skipped something, no, okay. Biodiversity sort of within our own cells, like body, the cells that are from bacteria actually are more than the number of our own cells, the concept of microbiome that is becoming more and more, feasible to approach given the genomics technologies that are out there. Obviously, it started long time ago by microbial ecologists looking at ecosystem and natural environment, and now we're looking at inside bodies and looking at our own ecosystems. And then I think, what I have realized, my background is microbiology, but I've moved into things that are much bigger, but they are also like macrobiomes. These are insects. One collection at the night in Costa Rica, you just put a net or like a light trap out there, and they just fly at it. And reality is that we don't have ability to understand these as a whole, as a community, but also there are very important scientific and socioeconomic issues within a context of something like this. Like, is this thing gonna be the vector of the next, disease that is going to be an important, disease to humans or animals that live there? Or is this going to, be, like the pest that is going to attack our crops? So these are the type of questions that you need to ask, directed questions. But then as a whole, how are these communities changing as a consequence of climate change or environmental change? And we can't just go in and pick one showy kind of big species and build all of our models based on that. We need to understand the whole thing. So far, I think this is probably the best paper that talks about the global indicators of kind of global biodiversity and the indicators of the decline. So many very good list of scientists have been working on developing this kind of meta-analysis. It was published in Science almost 2 years ago. And it's the data that you look at and you find out that it's really not data rich so much. What they have been able to get their hands on is like population of birds or, some of the red list species. And then when it comes to the aquatic environment, and I've been involved with aquatic environment, and, sort of, it's a pity to see, for example, that water quality, again, it's about biodiversity decline, is only based on physical and chemical quality. So there is really no biological data. So we're basically measuring the chemistry of water and trying to understand what's going to happen down the road to that system. And then when there are cases like this, like we really need to go in. This is the big spill, the BP, situation that, like happened. And then we have to go in and really find out what happened to that environment and the communities and what would be the consequences. This is the wetlands of Louisiana after, like the oil spill. And again, it's remarkable that, like the economic sort of value of a system like that is much perhaps more than the, the oil that was, like produced in the context of the production of it. And then we have a similar situation, but also in a more kind of chronic way in Canada. These are the wetlands of Alberta that are near the oil sands or tar sands, you know, and a number of other industrial activities that, again, you know, like we really don't know the impact of those activities on these wetlands, and some of them are very valuable ecosystem-wise. And then again, you know, like there is no difficulty finding, and this is probably close to you guys in a way. This was last year, you know, the ecological kind of time bomb as a consequences of what happened, you know, like as this, you know, like giant ship, you know, like had a disaster there. And again, you know, like we really don't have the ability to understand what happens to the environment when something like this happens, because again, we don't know how to measure biodiversity at large. And then agencies like Environment Canada, US EPA, I know there are similar agencies here in Europe that look at this as their job to basically understand the consequences of environmental change on the habitats. Like here is the Canadian Aquatic Biomonitoring Network that they use. Essentially, they track about 1,000 species of benthic organisms to understand the sort of the changes in the environment. They're like an early warning system. They're very sensitive to environmental change, much more sensitive and complementary to the chemical probes that they have. So they, and this is the type of organisms you get. You know, it's like you know, a petri dish of, you know, looking at a bacteria under a microscope, but this is larvae of the macro invertebrates that are used for this. Things such as caddisflies, mayflies, chironomids, you know, and those are the ones that are indicators of the aquatic health. And same thing happens in the marine system. We have similar organisms, so it's their early warning system. Now, this is the method that they use. Basically, the differential sensitivity is first, you know, you measure the habitat that is like a reference site, a pristine site, and then you characterize the community of the organism. So it's not like one species, but it's not everything that is in that habitat. It's an assemblage of organisms. And then you go and build a database of these reference sites and all the communities that are there. And then you go to your test sites and start looking at the test sites, compare them to the reference model, and then look at the other characteristics of that site, and then think about a predictive model and you know, like use that as your index. But the problem is that this biomonitoring approach has turned into a bottleneck in environmental assessment. And because it's very much done in a manual way, you know, like many other tools in biology that are very useful if you have the right people to go sampling for you and the right expertise to look under a microscope and try to characterize. This is essentially, this is Environment Canada's standard reference library that they use basically for the comparisons. And then if they want to do high throughput, this is a Stroud Water Research Center. They get a bunch of students with taxonomic keys, lots of coffee, you know, they put them in a room, lock the door and say, you have to identify all these bugs, you know, and that's the high throughput. This is the sort of high throughput way of doing it. And then you go and sort of look at the actual bottleneck. It becomes an identification bottleneck. So there is low capacity in the system that you see. There are usually less than 1,000 samples are looked at in a year within a context of a city or a stream. There is low throughput, like it takes a long time. The resolution often, because the characters that you look at are very, you know, limited, like larvae all look the same to me. And so you get to the family level, that is really, and I talk a little bit about this, that it's the power of your model to predict environmental change is really related to your resolution. And then it's done because of all these problems in a very low frequency every 3 years. Sometimes, you know, in a country like Canada, that we have lots of water and small population, it's not done very well. I mean, like there are sites, but they're not visited. And I mean, like the best places in the world are places like UK, that you don't have much of a natural ecosystem left, but there is a good biomonitoring, I would say, network. But in a place like Canada, it's very difficult to do it, and it's labor intensive. So nobody really likes to do this job, like, you know, for a long time. Now, I mean, okay, how science or how technologies can work, you know, towards making this system a bit better. And it's not a new thing, you know. This is kind of a journey that has started a long time ago, and others talked a little bit about that angle of it. So there are concepts and technologies kind of working together. Linnaeus kind of came up with a modern taxonomic approach, and then Darwin obviously put, the evolution gave the framework of classification and, linkages of organisms. And then technologies, genomics technologies like, Sanger. I just came from this campus. In fact, I was really interested because I've been using this image for many years. And then I was here having a meeting 2 days ago. So it was very interesting and historical. But then essentially, the work of Sanger and Kerry Mullis, the PCR. But then what's happening today, I think we are at a very interesting time now, because I think technologies are also giving us new concepts. The paradigms are shifting in biology as a consequence of things such as next-gen sequencing, and I get to that a little bit further. So it's like a kind of a circle, maybe, that we need to look at, like new frameworks, and even in phylogenetics, studying, and analyzing data, when was that maximum likelihood approach has been started to use, and not necessarily in biology, but in biology, long time ago, and when was that we got to start to use it in a real sense. And then again, we heard from Jason that we really have to cut corners still to be able to deal with a lot of problems. We can't really enforce the best conceptual frameworks in the real data. But again, I think it's a moving thing, and it's very good to see technologies evolving quickly so that we can, revisit sometimes these conceptual things. And biodiversity is being one of them that I am interested in. So the DNA kind of barcoding concept is really not, I would say, didn't just, occurred, and it's been going on. We've been using DNA to understand diversity or understand evolutionary relationship for a very long time. I think the paradigm that DNA barcoding kind of introduced is to standardize and use a minimalistic sort of approach for marker selection, and then go really horizontally wide and look at very large number of things. And again, to do that, we had to focus on a very small portion of the genome. In fact, a compartment of a genome from mitochondria specifically for animals. And then we have been focusing on a very small fragment of the mitochondrial genome. I argue that you can get different pieces of this, and the answer at the end would be similar, but you have to stop at some point, and you have to have a standard. I mean, this is a case with GPS. There are so many different standards for GPS, like Europeans, Russians got their own, and the one that was introduced and we are using today is just one, and I don't know if the best one or not. But if we couldn't converge on one system that is a standard, we couldn't have a GPS in our pockets. Same thing here, I don't think that there is any fragment especially for animals in the mitochondrial genome. There are lots of protein coding genes that give you the same type of answer. And this is the sort of type of thing that, we started looking at what is a species and what people really believe in as a species. And eukaryotic world, people are very attached to the species concepts that they use. In prokaryotics, there is no species concept, so that you can basically, use different types of cutoffs. Here, we looked at the assemblage of primates because we thought, okay, there is no dispute that, there are different species. And when you look at it, this is a very simple system. The genetics of mitochondria is simpler than, nucleus. And so you see less of a, variation within a species, although there are some species with more variation. And again, this is population genetics. But again, by and large, it allows you to differentiate members of different species, and you can live within a species variation. That's kind of the foundation of the identification based on this marker. And we started to look at this in a much more serious way by looking at groups that are very species rich. And these tropical Lepidoptera that we used for this study, they have been studied rigorously by professionals and amateurs for 250 years. I mean, they're very, very well-studied systems. So we selected these so that we are benchmarking this DNA-based approach against something that people believe in as a species. And again, I'm not saying that there are no surprises. There are surprises, but we had to start somewhere that, it's a rigorous test and is a solid taxonomy. And the answer was very, clear for us. We get about 98% resolution, and there is no other taxonomic system, that can get to this resolution. So DNA wins here, because it also allows you to basically use it as a framework and then bring in the specialist and focus on the cases that are controversial or you need other types of data. So nobody wants to define a species based on this. It's basically allowing a much more robust understanding of biodiversity. And in this case, like it was clear. And it's been on and on, like different studies, well over 1,000 publications that looked at different groups of organisms with this marker system. And again, there has been a lot of publicities and, media. And I think probably, the most interesting one for me was to see the cover of Nature with Linnaeus, with like outfit, like modern outfit, and then a barcode in his hand. So it's like, we are not diverging from the classical taxonomy. We are just giving new tools to taxonomists and other people like ecologists and other researchers to understand biodiversity. And again, there has been a pretty good kind of community effort behind this. This is the Barcode of Life Initiative, the first meeting that we had. Oops. Oh, you have to see this again. I'm sorry. So there are about 75 people in 2007, and I think, Felipe, you were here somewhere. You were in this meeting. No, you didn't come? Okay. So anyways, we don't have to blame you then. So yeah, the group that came together, they kind of felt after a few years of testing that this type of method has the ability to go further than just, like one lab, and like grad student work. It can become an international initiative. And there has been investment in our university. We've had, we just occupied the second building. This is the first building, the Biodiversity Institute of Ontario. Now we have a second building under the title of Center for Biodiversity Genomics. But, it's an extension of the same sort of movement. And there are several labs doing different things. And, we had the 4th International Barcode of Life meeting in Adelaide last year, and Philippe was there. And so it's kind of becoming kind of an international approach. And I don't think that there is so much of a forced international movement. Like genome sequencing, it's becoming more like every lab who wants to investigate, they go and build their libraries, but there are also centralized libraries and efforts. In terms of, other groups of organisms, this is fungi, and of course they are very cryptic and they're very important and unknown. And there has been a lot of efforts, like in terms of using markers for fungi. In May 2007, another marker, the ITS from a ribosomal segment, was selected. But this has been used since '91 at least, from the work of people in Berkeley early on. And then the cytochrome one works in some group, and again, but there are introns in the mitochondrial genome of fungi, so it's probably better to stay away from mitochondria. And then the other, like LSU, large subunit of the ribosomal segment, has been used as well. Now, nuclear protein coding genes. Again, in May 2011, it's back to ITS now. There was a paper, again, a community effort to introduce this as a standard universal barcode for fungi. And again, these standards need to be established so these communities go in and gather the data, in a kind of a horizontally and from different taxonomic groups. For plants, it's been a lot more difficult to get, to converge. It started from a marker from plastid non-coding region in 2005, then went to a 1 marker plastid of coding, then 2 plastid marker system. That was this paper, that was a plant working group, large number of researchers involved. And again, in 2012, they added another non-coding from nuclear marker to get more resolution. So again, these are community efforts to build a better system for identifying biodiversity. Now, if you look at protists, this is very interesting because protists are pretty much everything that is not a plant, a fungi, or animal. But evolutionary speaking, this is like an evolutionary tree of protists a few years ago. And, or the whole, eukaryotes, these are animals, these are fungi, and this is land plants. And the rest of them are protists. So how can you converge on just a small piece that can resolve all of them? And it's impossible. So I think that in protists, we have to settle on probably like a marker to get you to somewhere in this kind of diverse set of groups. But then you have supplementary markers, and sometimes you need to get more like a genome scan or something to get them resolved. Now, in wrapping this part, I just basically have a couple of points to make. First is that the DNA has been used extensively for biosystematics and biodiversity analysis. Projects like Tree of Life or Barcode of Life are kind of good examples. Now, we have learned that different genes are required to provide resolution at different taxonomic levels, but also like, you know, like we've learned that lesson very well from Tree of Life projects. But then we also have come to conclusion in a way that for different domains of life, to barcode them and identify them, we need more than one gene, and we need different genes. So one gene doesn't fit all. So we can't just basically go with one marker. And the other thing that probably is even more important to me is the way that we access the information. Like here I have some examples of, you know, like challenges. Like this is Suresh, my former postdoc, and he is basically putting all these bugs from like a jar that was collected, is separating them, putting them one by one to do the Sanger sequencing, the typical barcoding on them. And again, this is not really, this is doable when you want to build a library, but it's not doable when you want to monitor things and, you know, you increase the frequency. I mean, these protists, another example, you cannot isolate them. They're very small, so you can't use the typical sort of single specimen approaches. And again, that brings me to the next kind of topic of discussion is how much DNA information do we need and how to access it. And I think that, you know, like this is a fundamental question that we need to ask in terms of the genomics, and it's been asked, but not, I think that there is more to do in terms of the design and architecture of these systems. So here I have like basically raw data in a way. These are the alignment of variable sequences, and these are two different species. There are haplotypes of two different species, and these are kind of close related species. It's a tough case, but nevertheless, if you look at different positions, you will realize that, for example, if you look at this position 58, you can differentiate these two groups robustly using this position. And if you walk across, you will see there are several positions like this, although there is some other variation, but these positions can separate these two species. Now, but in reality, so do we need to get the whole thing? Do we need to get only a few of these? Or how much information do we need? And so that's a fundamental question that needs to be asked, I think, so that we can engineer methods that we can scale up. And we started to ask these questions because of another challenge that we were facing at the time. We were dealing with museum samples, and these museum samples are degraded, you know, so there is no large DNA for Sanger sequencing in like a bug that is sitting for 200 years, or like a fish in a, you know, a jar, you know, like sitting for 40 years. So we started to ask how we can design a minimalist barcoding approach to deal with the specimens that have degraded DNA. And then we started to make it like a universal approach, so a standard of that. And that happened actually in a time that, you know, like 2006, we were thinking about that and the newer technologies were coming out. So that allowed us to actually think a little bit differently in the context of using next-gen sequencing because there were short reads. Now, and this is the method that basically started it. The embedded technology, I'm not going to go through it because we had enough discussion about the technologies, but this one is interesting. The pyrosequencing chemistry was invented rather, you know, a long time ago, 96, and the parallelizing of it, I think, you know, in fact, I think that is the most important paradigm for me as someone who studies biodiversity, is that parallel kind of capacity that you can look at things, you know, like several millions of templates together. So in a way, for me, it's like running 1 million PCR and sequencing reactions together in one shot without the need for separation. And again, you know, like, but again, going back to how much information do we need for your question, we want to resolve the species, and we have a good database of, you know, barcodes already. So let's take a look and see which technology gives me the type of resolution I need for resolving a species. And at the time, Solexa or Illumina and Solid were not really feasible because the longest you can get you about 80% resolution, and 454 was the only one that can get you to the more than 95%, and Sanger obviously is sitting here. Now things are evolving, so Illumina 454 is here. These guys are better. Other technologies have been introduced. And this is the evolution of these technologies, the way that, you know, like we plotted it based on the diameter of the bubble shows the read length. And I think that where they stand here in terms of the y-axis is the throughput, and this is the kind of the progress. And so so you can see that there are kind of two ways of looking at it. Illumina has been more looking around like the throughput, solid as well, but then 454 is going after, like more read length, and again, like there are PacBio and others that are kind of intermediate in terms of their throughput. So, and again, but I think the most probably interesting part is that these are just the available ones that you see, and they are there. There's also all these other ones that we didn't talk about today, but they are coming. I mean, Ion Torrent jumped from here kind of to here, but there's so many of the other ones that are coming in, and many of them don't require amplification. I think the take-home message for me is that this is an industry that is evolving rapidly, and there is a lot of momentum around developing new technologies. So I think it's time to think about like taking advantage of this evolution in different applications. So again, like the concept of the industrial scale genomics that BGI started it. I was there, and one of our former postdocs is now employed in BGI, and so when you visit there, this is the floor with bioinformaticians. There are more than 4,000 employees here, and then they turned an old shoe factory into a genomics center, and they are basically building up rapidly and producing a lot of. This is the science and nature wall they have there. So genomics is moving into the industrial scale of data. It's becoming a big data enterprise, like financial sector, like many other sectors that are getting into the big data enterprise level, and that's one way of looking at it. What we have been doing is kind of a scale back, and we are focused mainly on developing specific technologies using these tools. And in this case, we kind of started environmental barcoding to deal with the biomonitoring problems. And this is what I'm going to talk a little bit about. So in this case, what we do, we get samples without separating them, like this jar of insects, water, soil, you name it. And then we utilize sequencing technologies, the next generation ones, like this is a 454, this is a MySeq. And again, we haven't invested in the genome center, because what we really essentially want to do is what my postdoc Shadi is doing, going under the hood and trying to break these and learn about them to develop new applications. So for that, we do need to have access to those tools. Once the technologies are built, they can migrate to genome centers, or they can go to the other places. So you get lots of sequence data from these environmental samples. You use some sort of a reference library or computational tools to come up with your species identities and sequencing counts. If they make any sense in terms of abundance, better, but basically that's more R&D. Now, but in order to build a technology like this, it needs to start from the scratch and beginning, where we go in the field and sampling things. A very interesting example is that when you talk to biologists who do field sampling, they go in and get their samples, and they use kick nets to get samples from the environment. They wash it off to go to the next site, and that works for their methods. But if you use DNA-based methods, you're introducing a lot of cross-contamination there. So we really need to go there and understand, like this side of it. The other part of it is that the sample size. I mean, this was a question that Ed DeLong asked me in a panel, said, what's your sample size for this? How do you determine how many samples you need to deal with the biases? That becomes, dealing in terms of, like the robustness of the workflow and the data that is coming out. And again, remember, there are lots of error rates, and we heard about those things in the context of next-gen sequencing to give you the real biodiversity information, not the bias. And then it comes to selecting your markers or sequencing length, and we heard about that, and a lot of bioinformatics. And obviously, I'm not going to go into any of these in detail, but one thing that is even more important to be able to use that is to have real-world environmental assessment programs to work with them in order to sort of justify or showcase the use of these new tools. And we have been doing some of this, but our first experiments started in our backyard. This is the city of Guelph. You call it a city or a town or pseudo-city. Anyways, it's not a very big city. And then there is a conservation area not so far from Guelph called Elora Conservation Area that we kind of started looking at these two sites as our experimental sites to understand this biodiversity through this system. And this is a published work that I'm not going to go through, but we focused on the macroinvertebrates, the typical group that environmental agencies use, and we collaborated with Environment Canada on this. So we compared morphological identification with Sanger-based barcoding or Sanger-based sequencing with this 454 environmental barcoding in pooled mixtures. So we put in things that we knew what they are. Because if you go to an unknown environment and get the samples, obviously whatever you get is very valuable, but it's very difficult to understand, the limitations of your method and to break it. So what we did, we looked at all of these in a mixture that we know what they are, and then went into systems like adults versus larvae and comparing unknowns in the conservation. And again, at the end we found this system has a better resolution in terms of giving you a species level, and with one sampling you can get more of biodiversity, although I'm not saying it's perfect, but again, we are comparing to what we have as our available method today. And then we went into sort of looking at much more complex things like soil. Here we're doing bacteria in this case with 16S and comparing the conservation area with the city. And then you need to do some interesting bioinformatics, and as others mentioned, you need to be aware and collaborate or do it yourself. We had to come up with this fast tree-based kind of rendering algorithm. There are about 80,000 nodes here, and so, but again, there are tools now being built. So, and then doing this, obviously, the value of this is that it will focus you on things that are interesting for you. Like you find this clade here that you see here, and it's not in the city, and then, okay, going after finding what they are and those type of things is the real power. And again, in the context of discovering biodiversity, I happen to work with Dan Jansen in Costa Rica, and I have access to all of his samples. So, I got this tube of flies, tachinid flies, that are kind of an important group biologically, but they're not very well understood and well known. So these black branches are everything that is available in GenBank and in databases, and the red ones are the ones that come to this collection, one collection event. So these are the unknowns of the eukaryotic world. These are the bacteria of the eukaryotic world that they are there, and again, who knows which one is going to carry something that might be important socioeconomically or evolutionary speaking, how do they evolve and those things. So the data is not available, and now we're getting a lot of data. And other very interesting examples, these are not marine, but we get to some marine examples at the end. So this is another one that we collaborated with a group of French scientists that have been going to Amazonia in Colombia and Brazil, and they're looking at land use and the environmental degradation as a consequence of different land types and uses. And they've been measuring biodiversity through traditional ways. That's very tedious. This has been going on for several years, and they've been looking at a number of groups of, and we get to start working with the insects, and in this case, Lepidoptera, these two groups, and we basically use this environmental next-gen sequencing approach. These are 454s, and this is basically the sort of phylogenetic profile of the community, all of them, and this presence or absence and the density shows, like the number of reeds, and these are farms, the sites in Brazil and Colombia. So this is really not a lot of sequencing. It's also not a lot of work. We actually get the samples prepared with the help of 2 undergrads over summer, and we did 1454 for this whole thing. We just barcoded and packaged them and ran them. And again, like, all right, one would ask, how do you know that this works? We go and compare with the morphology that they had done, and we see the same type of land use, landscape degradation trend that they find with morphospecies is shown in the DNA kind of bulk NGS approach. And in the context of the use and ecosystem kind of analysis, that becomes very important so that we can do these things very quickly, cheaply, using NGS. This is my undergrad, my grad student. Oh, undergrad, she would be very upset if I said that. She's been, working hard on this graduate program. So she's a graduate student in my lab, Jennifer, who has been looking at, we don't have many kind of big showy rivers in Toronto, but the Humber River is kind of the Thames, if you will, in Toronto, and it's going from, and I'll show you a map. So Jennifer has been going around and working in sites that already are used for biomonitoring using a collaboration that we built up with Toronto Conservation Authority, is the agency in charge of this. Now, these are the sites. This is the sort of system that we look at. This is the lake, and this river goes up here, and this is the city, kind of the urbanization part, and these are like sort of more sort of new neighborhoods, and then agriculture sites, and there are some conservation sites across this gradient. So it's a gradient. And these sites were selected again with the consultation of the environmental agency. Now, what I selected here is just one site because I want to get the kind of a detour and show you some of the technological tricks that can help you marry the old-fashioned method with the genomics method. Because if I put all of these in a hopper and put them in the 454, then later on, if I want to sell it and somebody says, how do I know this was not a pseudogene or some sort of abnormality, your sample is gone. So we have started actually utilizing the liquid phase to get the DNA without touching this part. That was started with an infamous experiment that we did to get a mezcal or a tequila and identify the worm from the tequila. That's actually what we did, and we published in Biotechniques. So basically, rather than trying to access the DNA from what everybody does, the tissue, we access the DNA through the media, and it also solves a lot of sample prep challenges that you deal with, what these bugs have, and the type of proteins that you need to deal with. So the results I want to show you is the comparison between what you get from this phase and that phase, and this study should come out, I hope, very soon. It's been under review. So this assemblage, again, is all the organisms, and as I showed you before, we compare. Here is the individuals one by one. We got them out, identified them, Sanger sequenced them, and put them here. This is their frequency. And then ethanol is the kind of media. This is tissue, 1 primer, 2 primers, 3 primers. And then this is the combination. And then you have the tissue and the ethanol, or vice versa. But anyways, the ones that are missing are very similar. So essentially, you get the same answer if you just go after your media and don't touch your tissue, because they've been sitting there for like a couple of days or something. And so it's a non-destructive approach to getting these samples. And this can be applied to marine organisms that you get. And again, like you want to keep it later. It's a type of things that you can do to kind of convince more like a traditional way of doing it, and also preserve your material for future. So this is the results. We get 100% of the taxa that have more than 1% abundance, and we get a little bit measure of biodiversity abundance using sequence reads. But I don't want to delve into this too much because it's still kind of early to make that claim. And then we go and look at the whole gradient. And this is the number of species that you can get basically if you look at that. And I don't want to make any kind of claims here because it's early, but the sites have been already studied, and they have been indexed based on their environmental kind of quality. And here, it's interesting, this site with 10 species, and this one with 10 species, they're in 2 different parts of this gradient, but they're both been really red, kind of grade red in terms of the environmental. I don't know what's happening here and here. Again, this might be a consequence of a farming, some sort of agricultural activity, and this might be a consequence of, like, of course, there's a lot of buildings, and, like this is a very dense part of Toronto. But, again, but you look at it, you come around here, you see, sites with more species. So these are the type of measures that we need to do later with this data. This is very early on, and Jen's PhD project is on this, and she's going to continue. We have done 3 years of sampling. We're going to continue to do that, and these are the 2 sites that I already told you. Another system is, again, going to tropics and understanding the diversity of these macrobiomes, using systems like malaise traps, and that's another sort of a study that is going on. These are 3 sites in Costa Rica, and we sampled them. This is like a Venn diagram of what you get. These are the number of reads that we got for this particular thing, and then we build the OTUs, or the species, if you will, for each of them, and these are the type of data that you can get. I'm not going to go into the detail of it. But again, we had to design new primers, kind of be able to kind of get the coverage that we need. And, this is the way that, like, it hasn't been possible to go to a place like a tropical forest, and look at these assemblages and understand their biodiversity at the species level with this kind of fast, frequent method. And DNA way of doing it with next-gen sequencing is pretty much the only way to do it, as far as I know, in terms of quick, frequent. And so we ended up getting 7,000 OTUs in these 3 traps together, and then we found that there are over 3,000 of them match more than 98% something in a sequence library. And so half of them has got some sort of presence in terms of the genomics space with annotation. And then from that, like you'll find about 9 orders of insects, 21 families. However, there are only 51 described species. So in a way, the difference between this and this is that, okay, I don't believe that there are not more than this described but there haven't been a library effort on those. So that's why, we hit something, but it's not like one of those described ones. So this process of annotating and doing the collections is very important. So another sort of way of looking at the data in terms of multi-marker system to look at biodiversity is that you do need different markers to get taxonomic coverage from different environments and different samples, but you don't need the whole genome or the whole transcriptome. Because again, you want to do everything that is in environment, and we heard that the metagenomics of doing it, it's very difficult, and building frequency data for that is even more difficult. So here we look at 16S, for example, and guess what it brings for you is the bacteria. And again, that's why you also have a lot of bacterial sequences. You can make a lot of inference. You look at 18S, you get more eukaryotes, and then you can 28S, it brings fungi, and then ITS gives you a lot of fungi. CO1 gives you a lot of unknowns because there are no libraries yet for them, but it gives you the animals and gives you species-level animals. And ITS and RBCL for plants or all green algae. So the combination of this should give you a good coverage taxonomically and the resolution-wise at the different levels. Now this was if you go with one marker, there are some challenges. Like 18S is a good overall biodiversity marker, but it's going to miss the species like crazy. So you see, this was a paper that came about 2 weeks ago that shows this widely used marker has the limitation of greatly underestimates the diversity in a survey of meiofauna. So if you do a multi-marker, I think you can escape situations like this. Now, in terms of some work that we have done on marine organisms or marine environments, this is the kind of northern Arctic, the Canadian kind of territories, and we got samples from several sites. We got ice core samples, and again, here in the context of global warming or environmental change, so we have 3 ice core samples that we collected, and then we have cold water samples, and you can look at the data very simple with you can unsupervised clustering algorithm, and these are your species or taxa, and these are your sites, and just by looking at this, we can see these 3 ice samples give you a completely different picture of diversity, and but that's not enough. We need to go further than that. So you can look at different groups of organisms, and you can, find interesting trends. So we have been looking at, here is the green algae portion of the data, and again, this is in context of a multi-marker system. So we have used multiple markers, and you see the picture is between the ice cores and these sites, and even among these sites, you see the variation. You look at another group, these are fungi, higher fungi, and you see the ice cores give you a completely different picture, and then these ones are different. And again, these are OTUs. The species here is kind of a soft concept, but these are OTUs, and you've done enough statistics to know that this is not a pseudogene situation or chimeras or things like that. And then again, you look at another group, Saccharomycetials, and you see a different type of diversity measure. And then again, I was interested to see the arthropods, and there are lots of them. Some of the copepods that are known to be in these environments, and again, the ice cores don't show so much of the variety of those, and so on and so forth. You can go to mollusks, annelids, cnidaria. All of these can be surveyed in one shot, and these are not really very deep sequencing experiments. But because they're focused on a specific markers, you can get a lot with a small sequencing run. And again, like this is moving from taxonomic diversity to functional diversity. I showed you the pattern with taxonomic barcodes that I showed you, but these systems, we also have been utilizing functional markers. These are functional markers for specific pathways. Rather than going and harvesting the whole metagenome, for a project like this, it would have been very expensive for us. Basically, we collaborate or we work with ecologists or industries. This is, in fact, in collaboration with the oil industry, and they're interested to know, for example, in terms of bioprospecting, in terms of new things, they know what they're looking for, and we can design, the markers for those, and then understand them, and then these are, again, your ice cores are here in this part, and these are the rest of them. So you can move from taxonomic markers to functional markers or functional barcodes, if you will, and use that. Now we're shifting gears. Last year we were able to get a project funded. It's a fairly large scale project and involves, ecologists like soil ecologists, plant ecologists, insect ecologists, or this is the officer of a national park that works with us in terms of sampling. This is the genomics people in my group, and some serious bioinformaticians involved, computational biologists, as well as environmental agencies. So this project that we kind of called it Biomonitoring 2.0, and the idea is to put all of these things that I told you in the context of an important, environmental challenge for us in Canada, which is, in a place that is very close to the oil sands operation. This is the oil sands area, and we have the Wood Buffalo National Park. I was embarrassed not to know that Wood Buffalo is actually Canada's largest national park, but is the second largest protected area on the planet. This is a very, very large piece of land, and it's very close to, so we have the second largest protected area on the planet, close to probably the second or third largest oil reserve together kind of in one place. So that's very interesting. And then, so we are using this to understand what's happening here, and kind of we have the ability to go, this is a national park, so the jurisdiction of the federal government, but we work with local, government and communities there, and now we are collaborating with industry. One good thing about this is that we have a lot of prior information, the classical biodiversity and ecological information, so that, again, we can benchmark our genomics approach, and we are not shooting in the dark. And the idea was to define what we call a biodiversity sampling unit. It's not just soil or water or, sediments. It's everything. So it includes soil and lots of repeats, you see. So we do 3 within each, and then we do 3, and then we triplicate that within 100 meters. So we can tease apart this within site variation from between site variation. And we do water and bentos and insects with pitfall traps and malaise traps. And we do it in 2 different regions of this park, one closer to this and one further down. And there has been a lot of, we didn't select the sites. This is Parks Canada and Environment Canada people that have been studying this system. And the idea has been to basically sample these things and use this environmental barcoding approach on them to develop the maps and then look at the models and the predictive models. Just recently, we got also permission and approval to really go to the oil sands sites themselves and sample as well, and hopefully that's going to give us a much better resolution. And this is, the way the sampling is done. These are very remote areas, so some of them you have to use helicopter, some of them you have to use airboats, and again, the traditional way of doing biomonitoring in a system like this is impossible. So it's the challenge, and hopefully the genomics will allow us to do this. This is my lab, the postdocs, grad students. This is kind of the picture we took recently, and some people are not in here. So, and they've done most of the work, and we're going to continue to do this, including a large number of collaborators, as well as Felipe. We have a couple of projects together that we're hoping to continue in this context, and the funding agencies. Thank you. Of measuring biodiversity as a whole becomes truly enormous. This means that when you move away from the species list, it's normal to think about like biodiversity a place or interactions, like plant defensive chemicals, trophic webs, or mutualism, and etc. So this has been a challenge, like we really have to focus even within the context of biodiversity. It's very difficult to understand it as a whole. Now, if you look at a diagram like this, like a word cloud of the definitions of biodiversity, what comes out is things such as the species, varieties, in the context of ecosystems, and then communities and variability and those type of things. The species sits in the middle, kind of the largest sort of object for biodiversity, but we need to remember in the context of ecosystem, and also looking at, we saw in the last talk that how population variation can determine some of the conservation efforts and how genomics can help that. So unfortunately, we haven't done a good job so far of documenting biodiversity. This is kind of what we know about biodiversity, about 1.9 million species of plants and animals. I don't even have microbial ones and some more like protists and things here, because we really don't know much about them. And even these, they don't come like in a central registry, like a library that you have all those. And in fact, I just before this, I was in EBI in Cambridge, and they were discussing projects like Catalog of Life, Species 2000, Encyclopedia of Life. So these are attempts that just started to document biodiversity. Now, perhaps a big challenge is even the difficulty in understanding or estimating biodiversity and different contexts. But the challenge, which I think I've been thinking about a bit, is how to use a map like this to monitor changes in the environment and the ecosystem over time and space, specifically over time and space. It's in a way, like we started having maps long time ago, but then we put them on a system that we can have in our pockets, like a GPS, and map positions of ourselves in the globe. So that's the idea. So the data generation aspect and analysis, in my opinion, needs to change so that we can use this to monitor changes. And that's where I think that next-gen sequencing technologies have revolutionized this field, and I'm going to talk about that. So what is biodiversity? There are so many different definitions. This is the one I like, that from Ed Wilson. Is, in a sense, is everything, every living thing, every living organism, and the molecules in them sort of represent biodiversity. But then a more pragmatic one is that I sent an email to my colleague, and I don't know if you see him. This is him, not the cow. But is Dan Jansen, the father of tropical ecology and conservation, and the one that actually kind of is coming from the same time of Ed Wilson, but he's been in Costa Rica and trying to conserve biodiversity. And I think his definition of biodiversity is a bit more complex and perhaps a bit more pragmatic. So the biodiversity of a place, country, county, habitat, ecosystem, a square kilometer, is the crude sum of all the species who live there and their genetic as well as morphology, physiological characteristics, and their interactions with each other and with their physical environment. However, of course, once you move away from the simple list of species, the task, we go a long way with Philippe, and I'm very pleased that you have this opportunity to work, in two different countries so very closely, and thanks to David and Felipe and all the crew to organize this, and Carlos and the rest of the team. And so what I'm going to talk about, as Philippe mentioned, is in a way is a continuum of what other speakers talked about, and it's very interesting. I don't know if you guys thought about that logic, but we are kind of getting more and more broader in terms of biodiversity and organisms, and I think we get ultimately to where diversity has not been very well understood in the microbial one in the next one. So I'm not going to talk so much about microbial ones because you're going to hear, but like starting from genomes and assemblies, going towards non-models, and now we're going to talk about like biomonitoring and biodiversity a little bit further. So I have I don't know if you see this, but this is a map of Earth at night, and I'm interested in urbanization and how it changes biodiversity. So if you really want to have a map of where things are in terms of urbanized areas, this is a good one because you can see everything. But this is more of an inspiration for me to see something like this and put it in the context of biodiversity. And so, and arguably, I would like to argue that DNA information can provide a map of biodiversity for us. And this is, as you have seen and heard, I mean, like, it's possible to use DNA to understand biodiversity in different groups of organisms. Also, there are very important scientific and socioeconomic issues within a context of something like this. Like, is this thing gonna be the vector of the next, disease that is going to be an important, disease to humans or animals that live there? Or is this going to, be, like the pest that is going to attack our crops? So these are the type of questions that you need to ask, directed questions. But then as a whole, how are these communities changing as a consequence of climate change or environmental change? And we can't just go in and pick one showy kind of big species and build all of our models based on that. We need to understand the whole thing. So, and so far, I think this is probably the best paper that talks about the global indicators of kind of global biodiversity and the indicators of the decline. So many very good list of scientists have been working on developing this kind of meta-analysis. It was published in Science almost 2 years ago. And it's the data that you look at and you find out that it's really not data-rich so much. What they have been able to get their hands on is like population of birds or, some of the red list species. And then when it comes to the aquatic environment, and I've been involved with aquatic environment, and, sort of, it's a pity to see, for example, that water quality, again, it's about biodiversity decline, is only based on physical and chemical quality. So there is really no biological data. So we're basically measuring the chemistry of water and trying to understand what's going to happen down the road to that system. And then when there are cases like this, like we really need to go, these are the type of estimates we have that are really rough. And in fact, recently there was a paper, I don't know if you noted, in PLOS Biology, that was a bunch of colleagues in, a group of colleagues in Halifax. They came up with a number, 8.7 million species. But I don't have that number here because I don't think that's so much important. What is important is what I think they mentioned in this code, that unfortunately limited sampling of the world's biodiversity has prevented to date the direct quantification of the number of species on Earth, while indirect estimates remain uncertain due to the use of controversial approaches. So we don't have the tools to understand biodiversity. And here is, like when you want to view, maybe I skipped something, no, okay. Biodiversity sort of within our own cells, like body, the cells that are from bacteria actually are more than the number of our own cells, the concept of microbiome that is becoming more and more, feasible to approach given the genomics technologies that are out there. Obviously, it started long time ago by microbial ecologists looking at ecosystem and natural environment, and now we're looking at inside bodies and looking at our own ecosystems. And then I think, what I have realized, my background is microbiology, but I've moved into things that are much bigger, but they are also like macrobiomes. These are insects. One collection at the night in Costa Rica, you just put a net or like a light trap out there, and they just fly at it. And reality is that we don't have ability to understand these as a whole, as a community,